All right, thanks so much. I want to start by thanking the organizers and thanking Alex for setting this up. I'm going to talk about the ultimate paper for my thesis work with Jeff Mumm, but also like to thank and congratulate the other two finalists as well. So I'll be discussing my paper, which was titled "A Large-Scale CRISPR Screen Reveals Context-Specific Genetic Regulation of Retinal Ganglion Cell Regeneration." We're going to breeze through some background, which is that we're going to talk about retinal tissue today, which is tissue comprised of 6 neural and 1 major glial cell type. Similar to Bridget's talk, actually, we're going to focus on a glial cell type, the Mueller glia in this case, which you'll see really have a profound impact in how the retina functions in the context of regeneration. And I want to note that the structure is highly conserved of the retina in zebrafish and mammals. Retinal diseases are typically neurodegenerative and usually occur due to the loss of specific cell types. So I outlined here the loss of retinal ganglion cells on the top of this diagram, which is a critical feature of glaucoma and some other diseases. Glaucoma alone affects 70 million individuals and doesn't have a cure. And there's also other diseases that are specific to other cell types, like the different photoreceptors. The reason that we're going to talk about zebrafish is because there's something very specific that zebrafish can do that we can't. And for the purpose of this talk, we're going to focus on the answer that they can regenerate neurons following retinal damage. So we'll use these kind of diagrams throughout the talk where these Mueller glia, these large green cells, following injury, naturally regenerate into all different types of neurons. So you see all 6 neural cell types here on the left. Whereas mice and humans and other mammals, the innate response to damage in the retina is just to form scar tissue and just to try and minimize the damage, but still you lose vision over time. And the focus of the zebrafish retinal regeneration field has been what we are going to call tissue regeneration, which is in response to a widespread injury. So from left to right, you'll see I've outlined this process as if you have damage to all retinal neuron types, which can happen from a light ablation or needle stab wound, the Mueller glia respond in a couple key phases of becoming activated and proliferating. And then you get the formation in phase 2 of these Mueller glia progenitor cells. And these cells are going to undergo neurogenesis similar to development, where they can then restore all different types of neurons. This process has been studied pretty heavily, and I'm highlighting a couple papers here that show we've already identified some of the key molecular factors that drive this process, which is very transcriptomically driven. You can see from Seth Blackshaw's lab on the left that Mueller glia specifically undergo a reactivation or an activation and reactivity to injury, including the activation of transcription factor ASCL1A and other genes, that then moves forward into a proliferative phase. Meanwhile, mice and other mammals have some reactivity phase with some similar factors activated and expression induced, but then go back to restoring rest. And this review on the right from Dan Goldman shows some of the factors that have been shown to play a role in the zebrafish response. We're going to highlight that ASCL1A is one of the key factors identified in both of these articles. And on the bottom right, the first paper that identified ASCL1A said that it was required for regeneration in this tissue regeneration context of multiple cell types. And another key point we'll talk about briefly is that if we learn about regeneration in fish and see what factors are turned on to naturally regenerate, the idea in the field is that we might be able to turn on these genes or stimulate mammalian regeneration by taking some of these insights. And we know that this is possible. Work largely out of the Tom Ray lab showed that if you overexpress ASCL1 in the mouse Mueller glia and inhibit histone deacetylases, you can get limited regeneration of some cell types. So the question for our lab in part is how can we better inform these approaches on the zebrafish side? From some work before this paper I'll discuss today, Jeff and I and others worked on a hypothesis basically to suggest that regeneration was going to be a very controlled process. And depending on the scale of loss and the injury I've got on the bottom here, it's going to impact the spectrum of specificity of regeneration, where widespread tissue damage, which are the models I was showing you above, would be high injury scale of loss and would lead to a high regenerative response of multiple cell types. But we thought if you titrated down the injury loss to certain cell types, you might get a more specific injury response, and that might be more relevant to diseases like glaucoma. So I'm going to show you data today where we damage retinal ganglion cells specifically in orange on the bottom left, and then trying to identify the steps that might drive regeneration of this process. And the key question we're going to come to outside of just what drives ganglion cell regeneration is how does this compare to tissue regeneration from cellular regeneration? I was fortunate to join Jeff's lab. He's a pioneer of transgenic modeling in zebrafish, where we create models that can induce specific injury to certain cell types. There's a transgenic model where the nitroreductase enzyme is hijacked from bacteria, essentially. And then metronidazole is given to the fish in their water, and it leads to cell death of the cells that express nitroreductase only. On the bottom of this figure, you can see that the yellow cells expressing nitroreductase get a 100 micromolar dose of metronidazole and then die on the right side of that image after 2 days. So we can really control this process now. And Jeff and I worked on this fantastic model we characterized and then performed molecular studies of ganglion cell regeneration, where we have specific expression of yellow fluorescent protein and NTR in the ganglion cells. You can see expression in the retina, where the cell bodies exist, and all the way down to the tectum in the brain, where the axons of the ganglion cells are sending information to the brain. So the first steps of my project were to characterize this model. You can see we used a variety of methods, including confocal imaging on the top left in both the retina and tectum, to show that 48 hours post-ablation or post-MPZ treatment, we can reliably kill over 98% of ganglion cells based on yellow fluorescent expression. And then after a week of injury, there's a pretty strong return of these cell types, about 30 to 40% by volume. And this gave us a baseline that now we can manipulate this process and see what might be good or bad for regeneration. I did some studies also on the bottom to show that the response of proliferation by the PCNA protein is in the muliglia. You can see on the right of the 48-hour post-ablation image that the large cells, which match the muliglia I showed you in the diagram that were in green, are strongly expressing this proliferative protein, and that this PCNA on the bottom right overlaps with glutamine synthetase, which is a muliglia marker. So we're getting reliable cell death, regeneration, and we know it's from the muliglia proliferating. I did a series of lineage tracing studies with EDU to show that the new cells that form following the MTZ treatment largely localize to the ganglion cell layer. So if you look at the second image on the left, to the right of the middle of the retina there, you see all these new cells in red that are showing new ganglion cells that have formed, showing that we do have a biased and selective cellular regeneration model. And I wanted to shout out Eric Horstick, who is at West Virginia, and helped us show that with MTZ treatment, we do significantly impact the vision of the fish, and that vision does come back over time with regeneration. And we're just showing data for the first few days after ablation here. Once we felt like we had a strong model, we wanted to sort of dive into the molecular factors that might drive regeneration. And we came up with a two-pronged approach that took up the majority of my remaining couple of years in Jeff's lab. The first approach is single-cell RNA sequencing, which we did with Seth Blackshaw, who was on my committee. And the question there was if we sequenced the retinas of zebrafish that got MTZ or didn't get MTZ at the larval stages at 12, 24, 48, and 72 hours, we could do 10x genomics and Seurat-based analysis to see what genes we were inducing in the zebrafish retina. And then key to the second part is a CRISPR F0 gene knockout assay, where we're able to inject one cell stage zebrafish larvae with 4 guide RNAs per each gene target that were induced by the single cell data. And then we could perform a regeneration assay where we ablate our control fish and our gene knockout fish, and then look on a fluorescent plate reader to see which gene knockouts accelerated, had no effect, or inhibited regeneration. You can see from my single cell data that we ended up getting plenty of cells. I think it was over 100,000 in total between all of our conditions and all our time points. The differentially expressed gene number is plotted on the right, where you can see that the muliglia, the red line, had a ton of differentially expressed genes at just 12 hours post-ablation, showing strong activation. And we have a little bit of a peak in the blue line, the ganglion cells, where damage is already occurring. So you can see how fast this process has already started. By 48 hours post-ablation, you see the peak of differentially expressed genes is now in the progenitor cells, which is going to encompass those Mueller glia progenitor cells. And then at 72 hours, you can see the strongest peak is now in the ganglion cells, which have now started to regenerate back. So we've got kind of a nice full process here of cell death and early regeneration. And one of the most important findings I think I'm going to share with you is that if you take all of the differentially expressed genes in our Mueller glia and compare them to the other injury models that have been published by the Blackshaw lab, you can see that there's actually a strong difference in how many differentially expressed genes are unique or shared between the paradigms. It was also interesting that both paradigms across their time points had about 1,600 to 1,800 total differentially expressed genes. And again, only about a third are shared. So we can already say that there's a huge distinction between cellular and tissue regeneration at the molecular level. My favorite part of this analysis was diving into this specific process I showed earlier, where I want to really say at the different stages of activation, what's happening. So to do this with the single cell data, I isolated the Mueller glia, the progenitor cells, and the ganglion cells to go along this axis of regeneration and conducted a pseudo-time analysis. You can see if you compare control in black in these middle graphs to ganglion cell ablation, that with the ablation, we get a strong induction of ATOH7, which is probably the single most important gene in ganglion cell development. Whereas a gene like ASCL1A is expressed at a higher extent at the early stages in these Mueller glia, but it's not by some huge margin. And then from left to right on this final graph, I show from the Mueller glia to the ganglion cell side, a series of genes that are induced expression strongly compared to the control, which includes commonly known activation factors like SOX2 from other studies, but also complement factor 7B, which is unique to this model. And then this middle and latter part of the graph show the neurogenesis factors that are controlling this process, where, like I mentioned, ATOH7 is highly increased, which was great to see and expected. But we also were a little surprised to see induction of some genes like VSX1, NeuroD4, that are thought to mostly promote other cell types. So we know that we're getting fairly biased regeneration, but we do see other cell types are still sort of being induced to some extent based on this data. The next thing we did was compare this data to an NTR rod model where we ablate the rod photoreceptors at 48 hours at the same time point. And you can see just in this one clip of this data that the ATOH7 factor is now completely not induced by the ablation, whereas it was so strong for the ganglion cells. And now ASCL1A seems to show a stronger inducement than the ganglion cell paradigm. So we talked about cellular versus tissue regeneration showing strong differences. Here we can see that cell type versus cell type, you still see pretty strong differences. And then the last part of my work was this very arduous CRISPR screen, where, like I said, we can inject guide RNAs per each target and then see what the effect on regeneration was. By the end of my experiments in the lab, I tested over 100 genes. About 2/3 of them were unique Mueller glia genes from my single cell data that I showed before. And about 36 or a third were coming from known genes in the literature, such as ASCL1A, SOX2. Some of these genes did result in developmental defects, which meant we couldn't test them in this assay because the fish wouldn't survive for this experiment. And you can see on this large log P and normalized YFP percentage that we have a series of genes in the top right where the knockout led to an increase in YFP percentage normalized to control, indicating better regeneration when these factors were knocked out. We have 7 genes on the left where we had an inhibition of ganglion cell regeneration, indicating that these genes in the normal state are required for this process. And then we had about 36 genes that I mentioned earlier. 35 of them had no effect or in positive effect on regeneration, the vast majority being in this no effect range in the bottom. You can see some imaging in the tectum on the bottom left of the graph to show that for neurogenin 1 knockdown, for example, we have a strong increase in YFP compared to the MTZ only condition at this early regenerative time point. And I want to focus now on a similar plot to what I showed before, which is that of genes that were known to regulate retinal tissue regeneration, there was 36. and we had 18 hits in my screen, there's only one gene that overlaps between these two groups that had the same effect as being pro-regenerative in both the RGC context and in the tissue regenerative genes that were known. I want to highlight lastly that ASCO1A was one of our strongest genes that, again, was required for other models, as in if you knock it out, you get no regeneration or very reduced regeneration, but we see strongly increased regeneration. This was actually super surprising to Jeff and I at first. We were convinced we made a mistake and did a series of repeat experiments to confirm this result, and it kept happening. So we decided that we needed to look into the literature to see how this could be explained, since our results were so in contrast to some of the field. If we look back at the literature, we found some data from the Tom Ray lab from the 2000s in the mouse model where they showed that actually in retinal development, ASCO1A expressing progenitor cells actually only lead to every other single retinal neuron cell fate. So ASCO1A retinal progenitor cells can make rods, cones, bipolar cells, and everything except for ganglion cells. And this actually started to make a lot of sense in the context of our data. So we wanted to now say, okay, we think that without ASCO1A, we might be biasing our regenerative process toward ganglion cells, but let's see if we can prove that with more data. So we did single cell multiomics on our ASCO1A knockout fish. And we saw that there was instantly gratifying results here, where if you just look at ATOH7, that gene I said is the most important ganglion cell developmental gene, with the ASCO1A knockdown on the top here of that accessibility curve, you have more accessible ATOH7. And then I constructed a similar pseudo-time axis on the left, on the bottom, from euloglia to the new ganglion cells. And comparing wild type to ASCO1A knockout, you can see that ATOH7, again, the expression is also induced. So we clearly have a bias here where we are getting stronger ganglion cell expression, pro-ganglion cell neurogenesis expression factors. We're also getting decreases and increases in other genes that are thought to regulate other cell types. So I want to highlight the one gene, NeuroD1, which is supposed to be good for photoreceptor development. You can see that that gene goes down as well in expression along our pseudotime axis. So we did an ultimate network analysis where we compared all of the differentially expressed genes in our ASCO1A knockout to developmental data. And you can see on the left here that the upregulated factors were good for promoting euloglia and retinal progenitor cell activity and even early ganglion cell lineage factors. But the more striking result was the downregulation of genes that led to other cell types, such as cones, rods, and bipolar cells. So it's not even so much that it's pro-ganglion cell, it's more like it's just very anti-other cell types. We summarize those results in the network graph on the right, where you can see that all of the factors on the right side of that chart that lead to ganglion cells, even amacrine or horizontal cells, were upregulated compared to the other cell types that were downregulated. And lastly, we did some validation results on the bottom left to show that the euloglia were not simply proliferating more following the knockout. So it wasn't just more euloglia were activated to lead to more regeneration. And with our final EDU stain, I want you to appreciate the wild type image on the EDU, where you see new ganglion cells in the left inner part of the retina, but you also see new photoreceptors on the top right of that image. And when we knock out ASCO1A, what we saw mostly was a decrease in these non-ganglion cell positive cells rather than an increase in ganglion cell positive cells. So to summarize, all this work, we created a new retinal ganglion cell regeneration paradigm that's relevant to diseases like glaucoma. We use this novel CRISPR screen approach that was published, I think in 2019, 2020 by another lab. And we found that there were several known regulators of regeneration that were just not needed for ganglion cells to regenerate, including that the loss of ASCO1A led to an enhancement in the fate biasing of ganglion cells. And we can conclude that cellular and tissue regeneration are very polar opposite processes, essentially. And this is a plastic process where we can actually modify this in vivo. And that strongly suggests that in paradigms like the mouse, where we're trying to stimulate regeneration, we might want to use these sort of fate biasing techniques to better model our therapies for diseases of interest like glaucoma or anything else. And with that, I want to thank the whole MUM lab and thank everyone that contributed to this paper. I copied this slide from my whole thesis presentation because this really was the culmination of a lot of years of work and help from a lot of people. So thank you. I'll be happy to take any questions.
Great. Thank you very much, Kevin. We have time for one question. I'll give people a few seconds to type their question in before I ask one. Okay, so I'll ask a question. So my question is about these progenitor cells have the capacity to form numerous different neuronal subtypes. How do they know which subtype is missing and to differentiate into that particular cell? Do you have some insight from your data?
That's a great question. One thing we really wanted to get more data for was microglia, which Bridget was talking about. And as she actually mentioned, it's hard to get those cells through single cell sequencing. But we think that that's a very important process where the Mueller glia that can also phagocytose cells and the microglia that also do that. And these cells are communicating very often. We think that that's probably the most important part of the process, where they can sense or pick up fragments of the dead cells and then sort of signal that to the Mueller glia to sort of start this process. So this work really gets to the later stages of that process, but I think Jeff's lab is now looking into the earlier parts as well. It's a great question.